Hi, as she said, I'm Kelly Hardin, and I'm really excited to be here today, and I'm grateful to have had the chance to meet some of you, and hoping to meet quite a few more of you today. But so I'm coming from Nationwide Children's in Columbus, Ohio. So I spent about 6 years working with Dr. Paul Martin on gene therapies, and so I've worked on GNE Myopathy and then some work on Duchenne muscular dystrophy as well. And so I just started my own lab at Mount St. Joseph University in Cincinnati, Ohio. And so I'm really looking to continue the work that I've been doing on GNE myopathy. And I'm really excited to be partnering with the NDF in some of this, and excited to be here today. So I'm going to talk to you about the work that I haven't done yet, but I am going to be doing very soon as I can get back into the lab, talking about a lectin staining biomarker for GNE myopathy gene therapy. And so first I want to talk to you some about gene therapy in general, because we've been talking a lot about gene therapy today. And so I really want to break down what gene therapy is and why it can be useful. So as many of you may know, gene therapy is the introduction of genetic material into the cell as a treatment, as compared to other treatments that may use different pharmaceutical compounds and et cetera. But in gene therapy, you're really delivering the gene itself into the cell. So when that gene has been delivered, then within the cell, it can produce the appropriate protein and potentially correct the disease. And so in particular, gene therapy is very useful for monogenic disorders. So these are going to be disorders in which there is a single gene that's mutated, and we know what that gene is. Because that's the case in which we can really do the most good in delivering a gene, because we know which gene is mutated, and we can then deliver that to patient cells. But the difficult part with gene therapy is actually getting the gene within the cell itself. And so that's why we need to use vectors. So vectors are gene delivery vehicles, right? They're allowing you to get the gene inside the cell where it can then be made into a protein. And so in particular, we found that viruses make especially good vectors because they're specialized for delivering that genetic material into cells. That's what viruses do all the time. And so we're taking advantage of that process in order to deliver the gene that we're interested in, GNE, into the cells. And I was telling you that viruses make especially good vectors, and in particular, in the past several years in gene therapy, we've really come to rely on one particular virus that you may have heard a lot about, AAV, or adeno-associated virus. And so many different viruses have the ability to deliver genetic material into cells, but the reason why we're so interested in using AAV in particular is because a few factors that make it particularly amenable to gene therapy. So the first is that AAV doesn't inherently give you any diseases, right? And so that's a big safety factor when you're talking about delivering a gene therapy. That gives an extra layer of safety. And then, as compared to other viruses, AAV is not particularly immunogenic. So it doesn't stimulate the immune system as well as other viruses to mount an immune response against the virus itself. So that's another benefit of AAV. It can also infect non-dividing cells, which is great when you're thinking about trying to get a therapy into muscle cells. It's great that it's able to get in those cells when they're not actively dividing, because it can get into existing muscle cells as well. And then finally, AAV does not need to actually integrate into the genome in order to express a gene and make a protein. So AAV can exist outside of the genome as what's called an episome, so without actually integrating, and it can express from there. And so that's actually really important because, again, it adds an extra layer of safety. Because if a virus were to willy-nilly integrate into the genome anywhere, then it could potentially disrupt another gene. However, AAV, because it exists outside of the genome and can express from there, it's great because it's not going to be disrupting other genes in your genome. So when you're thinking about what DNA actually goes into the virus, so obviously the gene is the most important part, right? In our case, it would be GNE that would be delivered into cells. But another component that you may have heard some things about is going to be the promoter that's going to control the gene. And so the promoter is basically is going to provide the specificity for where in the body that protein is going to be made. And again, that's going to provide an extra layer of safety. So if you are going to, if you're going to have a gene product that can be made only where it's needed, then that's going to be a safer situation than a gene product that's going to be made everywhere in the body. And so that's an important aspect of gene therapy, thinking about exactly what promoter you want to use in order to control where that protein is going to be made within the body. And then thinking about how gene therapy vectors are actually made. So in the last slide I was showing you, so a plasmid is just a circular piece of DNA. And so in order to actually make a gene therapy, you have what's called a triple transfection method. And so you have 3 different circular pieces of DNA that are all added into cells. And so the first is going to be the plasmid that contains the GNE gene. And so that's one component. And then you're actually going to add in 2 other plasmids that are going to encode other pieces of the virus. So for instance, the protein shell that surrounds the nucleic acid to make a virus is going to be encoded by these additional plasmids. So those 3 plasmids can be added to cells in a petri dish, cells in cell culture. And then virus can be collected from there and purified for a gene therapy. And then I know that Paul Martin's going to tell you more about different successes of gene therapy. But gene therapy has shown promise in several muscle diseases. And so this includes Duchenne muscular dystrophy, spinal muscular atrophy, and X-linked myotubular myopathy. And so those in early clinical trials have shown promise. And again, you'll hear more about that. But then more are in stages of development, and so it's definitely a blossoming field specifically for neuromuscular disorders. When you're thinking about the challenges in gene therapy, the biggest challenge is going to be delivery, right? Making sure that you're getting the virus into enough cells, getting that gene into enough cells to really make a difference. And so the good news there, though, is that at least for most diseases that we've studied, you don't need 100% of muscle fibers in the body to get the therapy in order to make a huge difference. So for instance, in Duchenne muscular dystrophy, the general consensus is that you only need about 20% of the missing protein in order to functionally correct the disease. And so you don't actually need 100% of the fibers to receive the gene therapy in order to have a huge impact. Again, that's going to be specific for each individual disease, right? So that's what it is for Duchenne muscular dystrophy. So when we're thinking about moving towards a gene therapy for GNE myopathy, it becomes really important to be able to assess at the molecular and cellular level how efficient are our viruses at getting into cells and how much of that gene product that we can see. And so as Monka was telling you earlier, first of all, it's important in the context of a clinical trial because you want to know as quickly as possible how well that therapy is working, right? And so in addition to functional outcomes, which are obviously invaluable in the context of a clinical trial, it's also extremely important to be able to visualize at the cell and molecular level whether or not your therapy is working. And in addition to getting to know more quickly whether or not a therapy is working, it's also really important to establish that relationship between exactly how much GNE you need in order to have a good functional outcome. That relationship between the cellular and the functional is extremely important. As I told you, a lot of work has been done in other diseases to establish that relationship, and so we need to do the same for GNE myopathy. So if you have that good relationship, you know how much GNE you need in order to make a good functional difference, that's great because then in terms of dosing future trials, you know what you need to aim for, right? You have a sense of how much GNE you would need to get back into cells in order to have a good functional difference. It's also important to validate those functional clinical outcomes that you'd see. So for instance, if you were working in Duchenne muscular dystrophy and you had a therapy that only put back, 0.5% of dystrophin, but you saw a great functional outcome, then that would be a red flag, right? Because previous studies have shown that you need more than that to have a good functional outcome. So it's important to have that relationship between the cellular and the functional so that you know that what you're seeing as a clinical outcome is really valid and you know what you're looking at. And then it also contributes to understanding of the disease in general, to have that sense of how much GNE you need to provide a good functional benefit. So Munkel was talking earlier about the difficulties in assessing GNE gene therapy, in particular because of difficulties with the antibody. So the approach that I am interested in taking is using lectins in order to visualize the enzymatic product of GNE, and I'll tell you a little bit more about that. So you've heard a lot about lectins today. So lectins are basically proteins that are going to bind sugars, okay? So lectins are going to very specifically bind different glycans, and some of those are going to be sialic acid. So lectins can help to detect the presence of sialic acid by specifically binding different sugars. And there are a couple types of lectins that I'm interested in. So some lectins are going to bind sialic acid directly, okay? So sialic acids normally cap the ends of sugar chains. So if you have a whole chain of different sugars, sialic acid is usually on the end here. And so if you use a lectin that specifically binds sialic acid, then if that lectin is conjugated or attached to a fluorescent molecule, then you can visualize where that sialic acid is. And so that's a really good strategy to be able to visualize the end product of GNE. But another good strategy is using a different kind of lectin, because some lectins don't bind sialic acid at all, but they bind the underlying sugar structures that are only exposed when sialic acid is gone. So some lectins are going to bind this underlying sugar chain that you wouldn't see if sialic acid's there. So in the case of sialic acid not being there, then a fluorophore-bound lectin would be able to bind this underlying structure and kind of show the absence of sialic acid. So both of those are really interesting strategies for kind of visualizing whether or not GNE has gotten into cells. And a lot of people have done a lot of really great work to show different lectins that are changed in GNE myopathy. And that's the work that I really want to build on. So some work has shown that those lectins that bind sialic acid directly are decreased in GNE myopathy, right? So if a lectin is going to bind sialic acid, then you would expect that lectin to be decreased, that binding to be decreased in cells of patients with GNE myopathy. But then the opposite's also true, right? If you have a lectin that binds that underlying structure that's only exposed when you're missing sialic acid, then that lectin you would expect to be increased in the case of GNE myopathy. As you can see here, as compared to an otherwise normal patient, muscles of a patient with GNE myopathy show an increase in a lectin that binds those underlying sugar structures. And actually, my own work has shown a proof of concept, and Paul's going to talk to you more about this as well, but a proof of concept that lectins can be used specifically to assess sugar changes that happen as a result of GNE gene therapy. So in a mouse model, I showed that when GNE gene therapy is delivered either specifically to the liver or specifically to the muscle, and then you use a lectin that specifically binds sialic acid, you can see after treatment that sialic acid binding lectin has increased in either the liver or the muscle as a result of that gene therapy. So this is a proof of concept that lectins could be used to assess what's going on after GNE gene therapy. So like I said, a lot of work has been done to show these changes in lectin binding that occur in the muscles of patients with GNE myopathy. And so while this has all been really fabulous work, one of the issues is that there are a lot of different lectins that have each lectin binds something a little bit different, right? A slightly different sugar structure. And if you look in the literature, there are a lot of different lectins that have been shown to be different in patients with GNE myopathy. So that binding is a little bit different. And some of them, the ones that bind sialic acid, are decreased. Those that bind underlying structures are generally increased. But the question is, out of this list of lectins that have shown to be different, which one is going to be the best for assessing GNE gene therapy? And so that's really what my new project is focused on, is going through all of these lectins that have shown differential binding in the literature and seeing which one is going to be the most promising for showing us a difference after GNE gene therapy. So the goal of the project I'm starting on is first of all to establish which lectin out of that list of lectins is going to be the most consistently changed across patients with different GNE mutations. Because as you know, there are many different mutations, and what we're interested in seeing is, first of all, which lectin is our best chance for showing a difference for the largest number of patients. Because if we're looking at a GNE gene therapy, we want to be able to assess that not just in a patient population with one mutation, but we want to be able to effectively assess that across all patients. And so the first goal is to figure out which lectin is going to be most consistently changed out of all of those that have been shown in the literature to be changed in GNE myopathy. And then the second goal is to actually deliver the wild type GNE gene, which is basically what would happen in the context of gene therapy, and then look for that lectin binding to be normalized. Not only do we want a lectin that's going to be consistently changed, but we also want one that's going to be normalized in the case of GNE being delivered. So to fix this first goal here, we want to figure out which lectin is consistently altered, right? And so in order to do that, I'm very excited to be partnering with the Lec Lab and with their new biobank initiative. And basically, I'm hoping to be able to test all of those lectins in cells with a wide variety of mutations, so we can figure out which lectins are going to be the most consistently altered across patients with a variety of mutations. Because again, with gene therapy, the goal is to be able to be as broad as possible. So we're going to obtain those converted myoblasts, like you just heard about. And then we're going to stain those myoblasts with the whole panel of lectins that I showed you, all of those lectins that were shown to be different in the literature. And so hopefully, as we compare those lectins in cells of patients with GNE myopathy to their family members, we can figure out which of those lectins are going to be the most consistently altered. And then obviously I want to quantitatively confirm that the sialic acid levels are actually changed as well as a backup measure. So once I have candidate lectins that are most consistently changed, I want to be able to make sure that that lectin binding is going to be normalized when GNE is delivered. So in order to do that, in those cells, I'm going to deliver the GNE gene and then test that lectin level with and without the GNE gene having been delivered. So the goal is that in these patient cells, as GNE is delivered and the GNE protein is made and then the sialic acid is made, that will normalize the lectin staining. And then again, I'll confirm that using a quantitative method as well. But the overall goal is to find a single lectin or a small set of lectins out of all of those that have been published in the literature that's going to be most consistently changed across the spectrum of GNE mutations. And then I also want to make sure that not only are they different in cells from patients with GNE myopathy, but also that those lectin levels can be shown to be normalized when that wild type GNE gene is delivered, which is what would happen in GNE gene therapy. Because that would give us a really good sense that lectin could be used to assess a gene therapy at the molecular level, so that we can really know what's going on and validate functional outcomes that we would find from a GNE gene therapy trial. So first and foremost, I want to thank all of you, because as you've heard, all of this work is thanks to the GNE Myopathy Biobank, which is from all of you. So I want to thank you for your willingness to participate, because that is just instrumental in my research and the research of many others. And then I want to thank the members of my lab, as well as my collaborators, and in particular the LEC lab, for providing that GNE Myopathy Biobank. And then I would like to give a huge thanks to the NDF for not only supporting this work, but for putting this whole day together. Thanks.
Thank you so much for that, Kelly. I have to say that on Facebook, you're getting a lot of oohs and ahs of people who go, finally, we've got it, we can follow. I think that was really, being able to impart it at that level is something even I overlook sometimes, what is gene therapy exactly? So thank you for that. Are there any questions for Kelly today, or was she so thorough that everybody understood everything? Yes, do we have a question over here? No? Yes? No? Did I see some hands? No? No? Kelly, you are so thorough. Oh, Monko's got a question.
It's not about thoroughness.
It's not about thoroughness.
Yeah, no. So, Kelly, I think the great thing about the biobank, and many thanks to everyone in the audience also that have contributed or will contribute, is that we also have family members who have contributed, who are carriers or heterozygous carriers. And do you have a feeling for what their lectin staining would look like? Would it be somewhere intermediate between a person who's perfectly healthy versus, patient? Where would, say, the carrier lie in that? And if you have some feeling for that. You might not, but I'm just bouncing the question idea.
Yeah, that's definitely a really interesting question. So what those levels would look like from family members who are carriers of the mutation as compared to patients. And so my main goal, right, is to be able to distinguish between patients who are affected by the disease and not. My guess would be that there would be some change in carriers as compared to people who are not carriers. But the way that we could really address that is through those isogenic cell lines that you were talking about. If we could have those patient cells, and then we could have the patient cells that are completely the same except for that GNE mutation. That would be really the best way to address that question of the different gradations of levels of sialic acid. Yeah, thanks.
Thank you for that. You guys, thank you to Kelly for her wonderful presentation. Please give her a round of applause. Kelly, thank you so much.